this video we will discuss about some of the practical aspects of the targeted therapy against cancer. So what the situation is like that when the patients that are treated by some of the targeted therapies, then after some time they get resistance against those targeted therapies. So in this figure we will discuss how these targeted therapies can be reversed by inducing this ferroptosis. Ferroptosis that we have discussed in our previous video. It is about the triggering of the program cell death because of the change in the iron concentration within the cells. So first we will understand how this uh, targeted therapies get resistance against the some of the inhibitors that are mentioned here in these figures with the blue symbols, and some of them we have discussed in one of our previous videos. You can refer to those videos, such as how this cetuximab functions. So in brief, what will happens when these receptors get uh, resistance against those kind of inhibitor. So these receptors will not be inhibited by these inhibitors, and they will trigger these pathways, such as these RAS pathways that will lead to RAF, then MEK, then to ERK, and then it will lead to this change in this epigenetic change, and then it leads to this epigenetic changes, and that will cause this tumor survival. Or along with this epigenetic changes, it will also helps in this DNA repair. That, for example, this olaparib in the ovarian cancer, it will inhibit this PARP that will prevent the DNA repair of the tumor cells. However, since this olaparib will since after the patients that are continuously treated with this olaparib get inhibited to this kind of reagent, so this its actual functions to inhibit this PARP will not function, and the tumor cells will be able to survive, as the DNA repair in the tumor cells will continue to occur with the help of this PARP. And the other pathways, such as this survival pathways through this P13K AKT pathway, through these VGFR receptors, will continue to occur, that will prevent the tumor cells from death. And this and one more pathway that will promote the tumor cell survival is this increase in the Ca2 amount in the cytosols, that will leads to this NOS formations, and that will promote this tumor cell survivals. So this is how the tumor cell survivals occurs when the receptors get saturated or resistance against the inhibitors after continuously uh, after continuous we can say after continuous in touch after continuous uh, interaction with this kind of inhibitors. So they modify themselves, or not just modify themselves, they somehow make themselves resistance against this kind of inhibitors. So how these things can be reversed? This thing can be reversed through their complementary inhibitors that will promote ferroptosis, as the reverse of these tumor cells is the ferroptosis, that is programmed cell death. So if this programmed cell death can be triggered, so people have found out a complementary reagent. So when this co-treatment of these two reagents are occur at the same time, that will lead to the ferroptosis or the cell death. For examples, in this olaparib, when treated with sulfazoline, then it will lead to the increase in the cysteine concentration within the cells, that will cause GSH biosynthesis, and simultaneously it will also this will prevent this GSS biosynthesis. This will prevent these combinations. This inhibitor sulfazoline will prevent this cysteine within the cells, that will prevent this GSH biosynthesis, and thereby the amount of GPX4 glutathione will get reduced, and that will lead to this ferroptosis, as the GPX 4 function is to prevent the ferroptosis. And similarly, the other inhibitors also somehow functions to reduce this GPX4. Uh, and in the case of this beta-alanine, it will increase the lipid concentrations or iron concentration within the cytosols that will lead to this ferroptosis. Whereas some of the inhibitors such as SH-GPX4 will inhibit this GPX4 concentration. So I hope you got an idea about how this reverse targeted therapy works. If you need to understand more about this reverse targeted therapy resistance using ferroptosis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.